I mean, we have till, I think it's 2 weeks to the presentation. We want to have it done.
Yeah.
We have this 2 weeks to now, do feedback stuff. And so we're fine. We're fine.
Good.
Yeah.
Yeah, because I'm at the medicine part of the project. There's so many different medicines, at least for our pathology, and it's all these different functions. I'm like, I can't do all these medicines. I'm just going to do the ones I'm familiar with, the ones I'm taking.
Well, and group things.
Yeah.
And instead of throwing a list at people, explain what does. You in your research go quite deep in an understanding of the pathology of what's behind, scene. You read the websites a little, and then you extract and you try to get the elevator speech that you can give them a package as full as possible, but not just rattle things. That's the difficulty, right? To make it so it's understandable. So in a medication, you can do it by classes potentially,
Yeah. There's 5 classes.
Yeah, I know, it's crazy. Yeah. Well, and then you can do which part of the pathology does it attack? What's the mechanism? That's interesting. Think about you explaining something complicated to a kindergartner that wants to understand it, or a young kid, or a person that is a little harder with language, or so, that you feel you don't, or a newbie, somebody like us who don't know all these technical terms. So that's sort of what the process is going to be. And then visualizing, the more visualizing you could do, the better. Or that's sort of what I do, try to do. And then you enlighten me and come up with this great way of doing things, and then I'm doing it that way. Well, I had last semester, in the spring semester, one guy, and he worked with Steve Jobs. I mean, he was a big deal back in the day. He's a little older. And he says, he just doesn't like PowerPoints anymore. It's just not, feels like this. It's just too, it doesn't connect to the audience necessarily. Those are things. So if you have a better way of doing it, take a board, make a good old board. You take it to class, and then we do it in class, or take a video of you doing it with the board or something. That's totally cool. It does not have to be a standardized format kind of thing. That's the beauty of what we have right now. All right, well, that got us right a little bit into the pathology thing, which is what I'm doing all day after I walked my pooch over here and took a nice little, have him take a shower, or at least the feet. It was rainy today up here, so that was nice. Any other questions since we're already getting into that, to the project or other things that are up that you'd like to talk?
I have a question about the project, if now is a good time.
Absolutely.
So yes, I think the 3 of us sent you our drafts, but not all in the slide form.
Okay.
Some just kind of in the research form, but we're going to meet on Wednesday to go over that part and put them into slides. So just in terms of when you would like that by, should we send it to you this week as soon as we're done, or do we have until Sunday, a week from yesterday, to do it?
I'm now is sort of the phase where I'm going through the things that come at me and give feedback. So it's good to send me stuff throughout. When you have stuff, send me stuff.
Okay.
Just upload it through you can always re-upload stuff, resubmit stuff through that portal. Or, depending, text me questions or things, and then we can have a little feedback going back and forth.
Okay, great. Yeah. And they're not on the call, in this now, but we're going to meet on Wednesday, so I'll update them also.
Okay. Yeah, and again, we still have time. If you started and you're really getting somewhere and you're starting to now create the PowerPoint or whatever format, visualizing things and thinking about how to present it. Now that you started on the absorbing of information, now it's the filtering and the figuring out how to present it, and that takes a little bit of time.
Okay. Yeah. Good.
But looking
Thank
at it,
you.
and the end goal again at the end, what we want is we want a 2 to 3 minute type presentation per person, a video, or we can come to the classroom. Again, we can do that this semester. For sure, we can always tag on an extra day. And I'm thinking, if you think about, I often try to do bullet point type of, thinking of either concepts or labels or things that I want to, ideas. And so I'm thinking, if I bring across one idea and put it a little bit in color, explaining or something, gets me about 10 seconds or something. It's about, thinking about it. And so about, then I start thinking concept wise, how deep can I go with something? So I'm thinking about 6 to 10 points a minute. So about 20 to 30 points in that time frame that we have. And that's pretty good to it's not easy to do that, but it's a good exercise to do that. And so if you start sending me things, then I'm starting to think in that format as well. And then we can brainstorm a little bit and have ideas.
Okay.
Again, text me too. If you have ideas and you want some feedback, text me that too. And we can I got my phone on all the time, like everybody.
Okay. Thank you. I do have just one comment that I'm studying with my dogs too.
Oh, that's pretty.
So there's one and there's the other. And studying with the dogs seems to make it easier, huh?
Yeah.
Okay,
I'm too nerdy and not grounded enough. I need this critter to put me on the ground,
Yeah.
Keep me organized. Tomorrow is hopefully his first work day where he comes with me to the practice.
Oh.
Today we're practicing driving, so we'll see how that goes. Children,
Okay. Yeah, I know.
So when we look at the nervous tissue, we're neurology stuff. We're talking about electricity going through the body and communicating the brain with the body and telling the body what to do. And so it's a pretty cool topic actually. When we look at this tissue, nervous tissue, it's its own tissue type. When we look at what is making up this brain and nervous system structure stuff. And when we look into the nervous system, we really end up describing two types of nerve structures, of nerve cells, cells, individual type of cells. We're talking as categories, we're talking about neurons, and those are the nerve cells properly that are responsible to do what we want the nervous system to do, which is conduct electricity through the body and talk to the cells. Basically, the brain talks to the body cells, and then the body feeds back to the brain and a little bit the spinal cord of what's going on in the body, and then we can make adjustments, sort of things. and besides those type of cells, those are the neurons. We also have helper cells, supporting type cells, and those are understood as glia cells or also neuroglia. Glia means glue, so those are the ones that hold the nervous system together and make it a cohesive situation that is also protected and stuff like that. For one of those nerve cells, those glia cells, let me see if I have more on them. I don't think so. No, one of the glial cells, for example, helps with nutrients, bringing nutrients to the nerve cells. The nerve cells need to be nourished quite extensively. Some of them are in the defensive system. They are taking part on the immune system type stuff. And other nerve cells are helping insulate the nerve. Since we're talking electricity, we're talking about You know, if you cross a wire, you feel it in your body. If you hold a live wire, you can have cross connections. You got to make sure that that's not happening. So the glial cells help with that, for example, as well. We're not going too far into the specifics of the glial cells themselves. I think here. No, there's 5 categories, and they do these different things. And we will touch on some of them a little bit more when we get to them, what they do. But I'm not going to make you study all the different names and all of that. I'm trying, what I'm doing here is I'm having my test review next to me, so which is actually something that would be good for you guys to do too. I want to make sure we at least touch on those points that are on the test that I want you to really understand. And so when we then go further into the neuron itself, into the structure of a neuron, this is sort of how we think of a neuron. A neuron is an independent unit, so we can study how the nervous system works on a neuron. That's what we kind of call when we call something a functional independent unit. So this is the smallest we can make something to understand how it works. Neurons are interesting. They are very diverse. They look very different from one another. There's different classes. Look at that. Looks totally different than that thing, for example. But we're going to study one that looks sort of generic. Neurons connect and transmit information through it, basically itself. It's like sort of a relay place where we can make a thought go forward or get stopped, or a piece of information. And so neurons connect to one another and pass along information. Or what they also do is they connect to muscles and make a muscle contract. And then the third thing they connect to are glands, and they make glands work. You can think of, sweat glands, for example. They work with glands. Also, the endocrine system, those are also glands. The endocrine glands, also, the neurons work with them as well. And so, but that's interesting to understand that neurons connect to either other neurons, to muscles, or to glands, and that's it. Because when we talk about anxiety, for example, I always visualize and think that, if I have an anxious thought and my brain goes crazy and the neuron just talks to the other neuron, I need to divert the thought somewhere else. So if I start using some muscles, I'm bringing it down into the body and decrease my anxiety. Of course, it's more complicated than that, but it actually works pretty well. Some people get sweaty and start smelly when they have an anxiety attack, for example. That speaks to the fact that the neurons talk to the glands. We talked about that. So when we get into a basic structure of a neuron, they all have these 3 fundamental parts to them. They have dendrites with sort of these 3 branches sticking out from this main part, which is the body, the cell body. We call the cell body also soma or perikaryon. And then we have a long extension going on the other side of that dendrites from going through the body, and that's the axon. And that's one extension that sends an impulse. So when we talk about the electrical impulse, a lot of this works with electricity. But electricity, we're talking ions. Ions are charged molecules. We talked about that in chemistry. And those are the molecules that are missing or having an extra electron on the outside shell, the valence shell, swinging around the core part. And so when these ions move around, well, then we create electricity, because that's what electricity is, moving this charge, moving the charge around. And you have all of that happening in this neuron, but where we really talk about an electrical impulse, we really talk the electricity, we're talking about that happening in the axon, mainly. That's where the nerve impulse gets sent. When you hit your funny bone, for example, by your elbow here in the middle, that's you hitting axons, you're hitting cables of nerves that then send, an impulse down to the pinky, if you ever had that happen. And that was a nerve impulse right there. So those are happening on axons. Again, if you have a question, just interrupt me, please. So that's a basic structure of how neurons are made up, what they're made up of. And then from there, we're gonna go to the synapse. We have classifications too, but I'm not really gonna test on the classifications. The classifications, to briefly work on it, are based on the numbers of extensions coming from the body. So we have some that have many poles coming from the body, multipolar neurons. Some that only have 2 poles coming out, and then some that only have 1 pole coming out. And those we call pseudo-unipolar because they're actually pretentiously unipolar. They were bipolars. They had 2 extensions, but then the body moved outward from this 2 extensions, and then there's only 1 extension here. It's really 2, but they call them branches then. It's really still and axon and dendrites at the end of the day, receiving and projecting ones. But this is done because we need to protect the cell body, and this is mainly a neuron type we find where the neurons, the cells come from the body into the spinal cord and go to the brain. And in that area, we have a lot happening, and we want these to be all collected together in a cluster, all these different extensions. And so what you actually have is you have many, many, many different neuron cell bodies that are holding together in a cluster. And they call that a ganglion. When you hear the word ganglion, it's not on here. When you hear the word ganglion, then that's what you're thinking of, cell clusters of neurons. When we get to the brain, we talk about that some more. But what is on the test is the synapse. And what is important about the synapse is that is a connection from one neuron, one nerve, to another neuron, or to a gland, or to a muscle. And essentially what we need to visualize is a fact that between the sending neuron and the receiving thing, neuron, muscle, gland, there is a space. And what happens is in a function of a neuron, a nerve impulse, that's these ions going around and back and forth. What they do here, they go across the cell membrane right here along the way. At the end of that axon, that's an axon right here, where the nerve impulse comes down. That's electricity. But all that really does at the end, it stimulates the release of chemicals into the space after that synapse, after that neuron. So at the end of that neuron, we have a little knob. They call it the synaptic knob, which is just a thickening. And in there, we have all these chemicals, and these are not electrical based. The release of them is electrical based, but the chemical itself is actually going to be the neurotransmitter, which we hear about, like GABA and acetylcholine and things like that. And those are chemicals that get secreted into that space from the sending neuron. And then on the receiving end, again, a neuron, another neuron, a gland, or a muscle, we have receptors, and they pick up that chemical. And then depending on what that chemical triggers is what happens on the receiving end. When you get to a muscle, the muscle will contract. And we talked about that muscle contraction thing with the calcium secretion, all that stuff in that chapter. And when you come to a gland, you're most likely secreting some hormones or some secretions and let them go into the bloodstream or out into the world, less in sweat glands, for example. But it's important to understand that the electrical impulse travels down the whole axon, and then at the end of the axon, we get to this thickening. In there, we have chemicals. They get then released. That the electricity is the impulse to release them, and then the chemicals get to a receptor on the other side, and the other side is another nerve, a gland, or a muscle, and then an action will happen. If it's another neuron, it either goes like, hey, we're going to stimulate the next neuron, or we gotta decrease the stimulation and inhibit it. That's usually what happens. You think computer language, you're thinking on or off switch. When you think electricity, you turn the light switch on or off. Well, you can dim it, but basically you feed electricity in or out. You don't have in neurology, you don't have different levels of gauging. You don't have that switch. If we want to grade how strong an impulse is, we do that by repeated firing. We send an impulse in rapid succession. That's how we code the strength of an impulse in neurology. That's the dimmer switch for us. What was I going to say with that? Oh, the look at the language too. Look at this. Always gets complicated when we do the language. We have a presynaptic neuron, or here membrane. Then at the end it's a membrane, and that's here. The neuron is here. Pre means before the synapse, and then we got the synapse, and here's they call that an intercellular gap. Yeah, because it's a gap between cells. That makes sense. It's just a language. It's just complicated. And then that's also known as a synaptic cleft or a synaptic gap. So all these words mean the same thing. So, just don't, try to get it into less words that you have to understand. And, because it can be complicated. It always made me mad when these words are all different, but it's just the way it is. And then the nerve or the area after the synapse is known as the postsynaptic. Here it's a neuron. It could be a membrane. If it's just a membrane, it could be the muscle or the gland. So that's the postsynaptic. So that's the synapse, like that, then a little thing. And then from there, we're going to have, what are we doing here? The discussion go into, well, the cool thing about understanding some of this stuff is you actually can use it for your own benefit, because you can start looking at neuroplasticity. See, the word plasticity means moldable. It doesn't just come from the Tupperware world or in plastic. And so the brain's very, very moldable, which is both dangerous, as in iPhone and phones, and very beneficial, because we can actually turn things around pretty rapidly and start, for example, think about how fast the mood can change. I mean, that's pretty drastic. And so that sort of speaks to that quickness. And so one thing that I really like as a phrase is that thing that neurons that fire together wire together. And what that means is if we send an impulse from one neuron to the next neuron, here, look, to the next neuron, to the next neuron, to the next neuron, over time, that thought is strengthened. And it is strengthened on a fundamental physical level by having a neuron that receives the stimulus, feeding back to a previous neuron and re-stimulating it. And by doing so, it strengthens that thought. That's why studying helps. And this stuff is fundamental. I mean, I was going to Bill Romanovsky's house, who is an old football player who still holds the record for most games played in the NFL without missing one. So he has serious techniques that he did, and he made, for some reason, he liked my work, and he made me go study stuff. And I did, and I was really studying muscles really, really hard. And one day I went for 2 hours, I worked them on the inner thigh muscles. Then I went home. Next day I studied for 5 hours on that stuff. I used to get up before to study this stuff because it's freaking, it's deep. And then you got to apply it. That's where it gets really deep. And then the next day I went back, and he felt the difference in my knowledge of those muscles with that one session of studies, because I created this understanding more and more and more and more. And so that was really interesting. I didn't expect that to happen, but he was like, wow, what did you do? It's like, I studied. But so that's an interesting understanding, because that means, and one of the things we're going to do with our health kit things, we're going to do a little bit of gratitude journaling. We're going to write down what, on an end of a day, we're going to write down 5 things of what, think back of a day and write down what made us happy today. Was it a sunset? And we then write it down. And by writing it down, we spend a little time thinking about it, reminiscing about it. and that sends off good signals in the brain to create this more strengthened pathway, and that over time helps us become happier. And there is this talk on the back of the booklets that I have, and I think I have it in the activity too, by an Austrian monk, and he says, "Want to be happy? Be grateful." And so that feeds back to this kind of thinking of then neuroplasticity. I like this picture too, where the pathway gets thicker and stronger. And then, of course, if we don't use it, it gets weaker. We understand that too, right? If we don't do something for a while, it goes away a little bit. Does that kind of make sense? Is that interesting to you guys?
Yeah. It's more complicated than bones and muscles, but
yeah, it is, isn't it? No.
Like bones and muscles, you can kind of just do memory, but this, I don't know, needs some kind of understanding too.
Well, it's the problem with the brain is it's like the brain is studying its own self. That's complicated.
Yeah.
That's just the way it is. And then when we get to the brain, now this is just the way it sort of works fundamentally. And to me, actually, this sort of like neuroplasticity stuff is ends up really being the applicable portion of it, and then the brain itself is a lot of like, what's this and what's this and what's this, and we slowly understand how it sort of works together. That's a little bit of the challenge with that topic, and that's why there is also hopefully a very decent test review, so we spell it out as well as we can. So then the next thing we looking at here, and let me see. We do have the nerve system is very delicate. The brain is encased in a skull, and if the brain gets inflamed, then that's a problem, because this inflammation means expansion, and the skull is not going to let that thing expand. And neurons are very, very very malleable, very soft. That's why they can grow, like the axons can grow. And so we need to be really careful of what do we feed the neurons. And so we have this thing called the blood-brain barrier. And we have these clingy glial cells. This is like one of those types, the astrocytes. They hang around the capillaries and cling around the capillaries that then connect the vascular system to the nervous system. And so what they do is they have these junctions. I mean, they have these little gaps that they let stuff through, but they filter. It's kind of a filtering system if you think about it. Yeah, that's probably an easy way to understand it. The blood-brain barrier is a filtering system. And then you can go forward and you go to the kidneys, and that's the next filtering system, which is exactly the same sort of setup. They're just not called astrocytes. Then they're called podocytes, which makes more sense because they're feet. But we get to that when we get to that, because the next thing that is important to really now starting to get to understand the bigger picture a little bit, and that's the sensory versus the motor nerves, the input versus the output. So now you're looking from a brain and the spinal cord's perspective, and you have those sort of be your centerpiece, your central nervous system. So that's symbolized by this here, and then also this this purple is sort of the spinal cord, the pathway going up and down the back, the spines through these vertebrae that we studied. And it's very well protected through the vertebra because it's a very delicate situation. And we have information that needs to travel into the brain and spinal cord from the body, and then get processed in the spinal cord or the body. And then we have to have information telling the body what to do afterwards to integrate that, after the integration, to execute something. And so we have this language that we use. We use the word sensory for input. So sensation comes inward, goes towards the information goes towards the brain and the spinal cord, and that's CNS stands for central nervous system. Sorry, I don't have that abbreviation spelled out here on this slide. And then once the information is processed and integrated, then we have a motor output, or an impulse going away from the central nervous system to the organs to tell the organs what to do. And that's the motor output, or nerves who carry that information. Or also we call those efferent, and the ones that come in, we call those afferent. And so the I look at in ascending, as in the nerve information goes towards the brain, into the brain. So you get all these terminology that the books are using, so we got to cover it. And then the motor, the brain telling the body what to do, that nerve energy is in the motor nerves. You think in the muscles contract, that's motor energy, you know, the motor of the body runs, you can walk when that happens, sort of thing. And that is also known as an efferent nerve energy, or output, input versus output. And then when we look at most nerves actually in the body, in the system, they're gonna carry both of these energies, the sensory and the motor part, and they're called them the mixed nerves at that point. We have very few actually that only carry one or the other. One of them, for example, is the eyeball. The seeing nerve only carries seeing information into the brain. It doesn't have an output, unless you're some kind of a superhero, then maybe it does, but not for most of us. The smell is the same thing, or the taste kind of thing. You only have an input nerve, but most of them are both. So if you actually, when you hit the funny bone, you hit the funny bone and you feel info going by sensation, but you're also gotta have to, if you hit it really strong, you have some motion. If you have some motion happening, then you're gonna have some motor. Otherwise, you're hitting the sensor because you're feeling what's going on in the brain. Even if you feel at the end point what's going on, if you hit it here towards the brain, you're still sending the information into the brain that tells the brain what to do. That's another very, very good point that might be a little tricky. Make sure you tell me if you don't get it, because it's more an application point. The nerve, the sensory nerve has, or every nerve has, a receptor type thing, an information picking up type thing end. And then it has on the other end a giving, an output end, where it sends the neurotransmitter out. That's the synapse that we discussed. The thing is that the rest is just a cable. The rest are only cables. And so if you compress a nerve anywhere along the cable, the brain's still going to think, or the place where the synapse happens, which in an input is the brain, mostly in the spinal cord, that only going to think that the end point of the nerve gets stimulated, because the brain only understands that the receptor is stimulated when there's an impulse coming in. It doesn't understand it's like anywhere along the path it could be compressed. It doesn't get that. It's kind of like an electrical signal. If you cut the cord anywhere along the line from the outlet to the computer won't work anymore. It's not just like the end of the cord doesn't work anymore. It's the whole computer doesn't work anymore. So that's kind of an interesting point. If that's a little too difficult, we can talk about it some more later. But from an applications perspective, that's very important, because if you think about somebody has carpal tunnel syndrome and has carpal tunnel syndrome and feels the carpals get all fired up, you can have a compression of the nerve, not just at the carpal where it hurts, but all the way along the pathway where the nerve travels into the brain. And so they actually test that these days, and very often it's in the forearm where the problem is, or it's in the neck where the problem is, for the carpals that cause that sensation, that pain or tingling type thing. Just is nerve covering. So I already briefly talked about that the nerve has a covering around it. That's one of those glial cells, and what that one does, it makes the nerve go faster because it insulates it, so it doesn't cross talk to other electrical wires. But you don't just have a nerve covering around each individual nerve extension. You also have bundles of them get connected together and create what's called fascicles or fasciculi, as a plural too here, fasciculi. And then we also have coverings around the whole nerve. And so if you cut a nerve out of the body, you're gonna get that whole nerve. And then when you start, well, it's too small to really dissect and see inside unless you take magnifying things. But that's how it looks on the cross-section side there. And then from there, what I want to touch in is the fundamentals. And when I look at how the physiology kind of stuff, how the whole thing works is a nerve impulse. The first slide here, we kind of talked about that already. We talked about the brain and spinal cord. It's just a better picture of how that's the central nervous system. And then all the nerves coming out of them, these other yellow things here, these are all called nerves. And these are all together considered as the peripheral nervous system, as the center and the periphery, the outside. And the basic functioning of every nerve is sort of what we talked about with sensory and motor. But before that, you have a receptor, and then you have an input, a sensory input, and then you have an integration or interpretation, and then you have a motor output, and then at the end you have that effector, what happens with a nerve muscle or a gland. So that's just spelled out again on here. This could also easily be seen when we talked about the negative feedback loop. It's the same kind of concept. Info in, info out. Well, info in, integration, info out. And then the rest of this chapter is pretty much about electricity. And so when we look at the nerve impulse, let's make sure we understand most of a nerve impulse is based on this electricity concept of opposites attract and likes repel. The magnets that snap together and pull apart is kind of that same concept. And so that's when we get to the positives and the negatives. And so that's how we create nerve impulses. And then in the body, what we got to do is we got to move these things around to create these imbalances, so to speak, of these positives and negatives. And that's what creates a nerve impulse. And so the other thing we need, and we got to balance these in and between the cell membrane in and out of the cell. And this is like on the axon, like along the whole tube, the cell membrane. And the other thing we need with electricity, we need a boundary. We need to make sure these ions can't just flow around all over the place. And in the neuron, we got the cell membrane as being that boundary. That's an oil film. And oil repels vinegar, and vinegar is watery, and ions are watery. So that's hydrophobic and hydrophilic stuff. And so the ions cannot cross the oily film of the cell membrane. So we can play with that as concentration gradients. How many positives do we put to one side and how many negatives do we put to one side? And since those are carried on chemicals, we can play with going to chapter 3, chemical movements, the concentration boundary or gradient, concentration gradient. So we can say, oh, we're going to have a whole bunch of sodiums on one side and not on the other side. And all of a sudden, we open some channels that let them flow freely, and all the sodiums want to flow into the other side and balance out. And the funny thing is the sodiums here, which are causing that nerve spike impulse, carry that positive thing with it, and that positive thing creates that electrical impulse. And then we can measure that with voltage, and voltage is the difference between positives and negatives from a side to side with a boundary. And here, this is a cell membrane boundary. And so we can measure that, and then that's that change in voltage creates that impulse, that nerve impulse. And so then we can travel that impulse along the whole pathway of the nerve, and it travels in electrical signals either towards the brain or away from the brain. And so that's sort of on the fly of how that whole thing works. Does that fundamentally make sense to you guys, or is that leaving a whole bunch of holes there?
This was confusing to me.
Okay, we're at. Then let's go back. What part? Do you have an idea?
Yeah, it was the graph where you're trying to, I guess.
Can we go further?
Further, yeah.
How was it so far? What I mentioned, how I explained it, was that all right so far?
Yeah.
Okay. Because then we can get to the graph.
Okay.
And
Because I'm like, was this what you were trying to show in the pictures?
Yes.
Okay.
So the graph, this is explained a bit more detailed, this graph. See right here? This is what I explained just now. What goes on here? Can you see you got the graph on the big picture?
Yes.
Okay. So the ions moving across the cell membrane is how we create electricity. So the ions are molecules, and they're charged though, so they carry a charge themselves, and that charge creates electricity. So ions moving in and out of the cell along that tube across that membrane creates that electrical charge. Does that make sense so far?
Mhm.
Okay, good. To all of you guys, Andrea, you still here? Or we just us?
Oh no, I'm sorry, I'm here. I mute myself. Yep, all good.
Okay, good. And then the graph simply speaks to who is going to travel across that cell membrane and what does that mean. Okay, so as we start here, this is when we are resting. This is when we're asleep. On the nerve is asleep, nothing is happening. And then as we want to send a nerve impulse, and maybe I should back up. When we talk about a nerve impulse, we're talking about specifically what goes down the axon. We're not really talking about what goes on here, even though we're still exchanging ions and creating little electrical echoes, so to speak. But when we're really talking about a full nerve impulse, we're talking about what goes on here. And then this is the spike that we can see. And so as soon as we have enough of a stimulus in this area here from what the nerve receives from before, and then it says, hey, we're going to send a nerve impulse. So at that point, we make a decision. The neuron makes a decision, says we got to send an impulse to tell the next thing what to do. And at that point, that's that action potential. And so an action potential is the same as a nerve impulse in our regular language. And at that point, that's what's happening here on this along this tube. And the first spike happens when the cells, the cell membrane has receptors. It has channels that open for sodium channels.
Oh.
They open. When we trigger them, they open. And when all they open, we're going to get all this sodium coming from the outside into the inner space of the tube, bringing all this positive energy with it. See the positive ions. And then that change here is measured by the ions bringing that positive charge with it. That's voltage, right? And so the voltage all of a sudden goes from negative, this is negative. I don't know where the negative went. Negative 55 over to positive 30. And that's the nerve impulse that we feel. So far, so good, or not yet? Not anymore.
Get it.
And then at that point, the sodium channels close. I think they are closing because the voltage changes. They feel that. They have a trigger. And then at that point, the potassium channels open. And so when we go back to the space here before we start the nerve impulse, we want to go to this picture back, and this is sort of at rest. When we have a lot of potassiums on the inside of the membrane, and a lot of sodiums on the outside of the membrane. And we're doing that by continuously pumping sodium to the outside and potassium to the inside. And so that's called the resting membrane potential. And so that's necessary before we start this process. So molecularly speaking, chemically speaking, we have a lot of sodiums on the outside before we start that nerve impulse, and a lot of potassiums, the Ks or potassiums, on the inside. And then when we start that process and we just open the channels passively, all these sodiums come in and bring that positive charge to the inside and spike it. And when it reaches this positive 30, more or less-ish, we're not going to be hung up with numbers too much, then the ion channels for sodium close and no more sodium can come in. But instead now, the potassium's open, and all that stored potassium on the inside wants to go out. You visualize the Tokyo subway, and they push the people in, and at the other side, they just fall out. It's like that's that gradient, that concentration gradient. And all the positivity that's attached to the potassium, still positive, will flow from the inside out to the outside of the cell tube. And that's known as repolarization. And all of a sudden, when we then measure the inside of that cell becomes more negative again, because we're taking the positive away from it. We're taking it to the outside. And then we're back to normal. Does that make sense?
Yeah, kidding.
Does it come in slowly? It's okay. It's complicated because we have chemicals and we have charge. It's two things. The chemicals move with the concentration gradient, and the charge is attached to it and creates the voltage difference. So first, we carry more positive to the inside to create this spike, this change on the inside. And then afterwards, we carry positives back to the outside attached to a different chemical. But it's still positive charge. And then that brings the charge back down. Getting clear?
Better.
Okay. No, it's complicated, but once you get it, you get it. And then everything comes from that. So that's not, it's okay. This is the complicated stuff. How about you, Andrea? How are you doing? And anybody else? I'm not seeing who else is in.
Yeah.
Any add-ons?
Yeah, I like the Tokyo subway analogy.
Yeah, it was funny. I was there, and it was really a funny event.
Yeah, no, I
Unexpected.
wrote it too several years ago. So that helped me. Again, picturing little stuff is harder than picturing the bigger stuff. So I guess that's why I like the subway analogy.
Yeah, so it's that concentration is understood with maybe that subway analogy when it's bigger things than compared to smaller molecules. And then the other thing is important to understand is that I think is that gradient of the chemicals, how they're always stored more on the outside of the sodium on the outside and potassium on the inside. And that is done by a pump, by the way, that causes ATP. And the cool thing about that is you want events in the body that have to take place rapidly. You want them to be passive events like this kind of stuff. Setting them up is okay to have active done with the pumps, moving these chemicals across so we can reestablish the chemical environment so we can send the next impulse. But the impulse itself has to be a passive event so it can be so fast. And that's kind of gets confusing there. Well, let's we can also meet offline again, Vinny, or anybody else who's listening. Just shoot me a text, and I'm very happy to meet anytime, pretty much, when I'm not with patients. Okay.
Okay.
And if questions come up as we keep talking, interrupt me and ask them, please. Very important, because this is the kahuna. Everything else is easy peasy. And then here, there's one more thing. This hyperpolarization is this divot here, and this is simply to do with the fact that the potassium pumps are slow to close, and so they send a little too many potassiums to the outside. I mean, it's not too many. The body obviously uses that to its benefit, and it's just in a period where you cannot have a nerve impulse firing again, which you can anyway, because you need to restore the chemical environment. So again, the chemicals move and the charge moves with the chemical, and the charge causes the electrical impulse. It's kind of this two-step process kind of thing. And then we already talked about it that, once you send a nerve impulse, you can't stop it unless you cut the nerve in the moment, but that doesn't really happen. It's too fast. But how do we code a strength of an impulse? Is the voice loud or is it soft? And that's coded by frequency. How fast does an impulse get shot after another? And so that's important to understand and not that difficult, I think. And then the last thing I want to talk about that's really, really cool is the fact that what we just talked about happens all along here. But if it happens all along here, it can be a very slow process, even though it goes fast in terms of how we explain it. But, you know, these nerves can be from your big toe to the brain. That's a long distance. And so what the body does that's really cool, it wraps a layer of insulation around part of the axons here, or the cable, and then leaves a little gap, and then wraps another, you know, insulation thing, which is plastic, which is a fat layer. See here, you see the different layers. It's just wrapping a cell membrane around itself. And then what happens is, the cool thing is, along the where it's wrapped, these ions cannot exchange, so they cannot make a nerve impulse. But the pull is strong enough that the impulse goes from where there's a little gap to a little gap, and it ends up jumping. And we call that a saltatory conduction. Where's the word? So here, look, saltatory conduction. A really cool word. And it's done by these Schwann cells, which is also cool. The Schwanns, Schwann cells. And the other thing that's a cool word, but you don't have to worry about the word, is the nodes where the electrical thing still happens. They call them the nodes of Ranvier. So that's just, you know, a word if you're an anatomy nerd like me. But the process is kind of cool, and that greatly speeds up a nerve impulse, because nerves travel different speeds. A nerve that needs to make sure you don't trip over something is a very fast nerve, because you don't want to trip. And then the muscle reaction is also very fast. But a pain nerve, as we all know, is a very slow nerve, and is grinding down because pain means it's danger. We got to pay attention. Now, of course, in our society, we pop an aspirin, which is maybe all right if we don't do it all the time. But basically, the pain nerve tells the body something's wrong. We got to change something. And the more we listen to that, so that nerve energy travels very slow. And continuous. The other thing that makes a speed go faster or slower is how thick a nerve is. So the diameter of a nerve is also very important in speed of a nerve. All right, so that's pretty good for that. So with that, how are you guys? Any questions? You guys had enough, or you want to tackle the brain a little bit, or you want to get through it first yourself and then we go through it?
Well, I have another meeting at 11, so I'll tackle the brain first.
Okay. Well, and it's, you know, the brain itself really is now, once you hopefully understand some of the foundation of how it works, a little bit of the in and out thing, you know, the brain spinal cord periphery. Then when we get into the brain, it's really a lot about what part does what thing. You know, okay. So start with that kind of thing. Have a first read through it. You know, the different lobes, what does what, and then from there, what we can do is, once you have a first go through it, we can pick it up next week and go through the brain more in detail and have questions.
Okay.
That sound all right for you guys?
Yep.
I think that's what we're pretty much doing this week. I didn't even know what else we got this week. Where is my Safari? Oh, yo. What do we have? What else do we have this week? Do we have the gratitude journal yet? I don't think so, right? Let me look real quick. Sorry, I was focused on getting into these pathology things. No, we're starting the pathology. We're starting the gratitude journal. So I'll finish up with that real quick, Vinny, and everybody else, and then we can go to questions. But the the gratitude journal is very simple. I already mentioned it. What I want you to do for 5 days, I want you at the end of the day, write down 5 things that you appreciated that made you happy during that day. And then reminiscing in that thought for a few moments, and then go to sleep or something like that. And also watch this 14-minute video. He's really funny. And then if you have any questions from that, it's a simple activity. It's not too complicated from doing its perspective. It's more like one of those where you want to just do it for a few days to get a little feel for it. And that's about it. And then we just do a little blood pressure discussion, but that's no biggie. We're used to these discussions now, I think. I really appreciate reading them, by the way. It helps me greatly motivate me keeping them up. So, but that's it. What I have. That's all I have.
Okay.
Thank
We
you.
have coloring, Andrea.
I know, which I love.
Hey, I know it's a good activity because it is a lot of stuff, but at least it slows us down a little bit.